We are still on the topic of DNA, but in Module 8, we are going to be learning about the inheritance of traits from parent to child. You will need your Module 8 graphic organizer in order to complete the notes for this presentation. This can be found at the end of Module 8 in your student handbook. Up at the top of your graphic organizer, you will see a place to write in a definition for the word genetics. Genetics is the study of heredity or the inheritance of traits. You will also see a place to write the name for the person who we call the father of genetics. His name was Gregor Mendel. Gregor Mendel was not a trained scientist. He was actually a monk. He lived in a monastery and worked and did some experiments with pea plants in a garden, and this work with pea plants allowed him to discover the basic principles of heredity that we still know to be true today. We're now going to record those 3 principles of heredity in the space provided on your graphic organizer. The first principle of heredity is the principle of dominance. So don't only write in the word dominance, but go ahead and write the definition for that as well. One form of a trait can hide or mask the other form. You will see in the chart some examples of this in pea plants, which is how Gregor Mendel did his work. Tall is the dominant trait. Short is what we call recessive version. Round seeds is the dominant, wrinkled is the recessive, and so forth. The dominant traits, when they are present, can hide, mask, cover up the recessive trait. Mendel's second principle of heredity is the principle of segregation. You need to record this on your graphic organizer as well as the definition. Each individual has 2 alleles for a trait which are separated when the gametes are formed during meiosis. Now this can be kind of difficult to understand, so it's important that you take a careful look at this diagram. In the first cell at the top of the diagram, you see the parent with 2 chromosomes, one labeled with the big A and one labeled with a little a. These represent the 2 alleles in that parent individual. When the gametes are formed, gametes are sperm and egg, and they are formed during meiosis, those alleles are going to separate or segregate. Segregation means separation, so that might be a word that you write down to remind yourself. Some of the gametes, as you can see in the diagram, are going to receive the big A, and some of the gametes are going to receive the little a, meaning that the parent can pass either of those alleles on to his or her offspring. Now you need to look at your graphic organizer and look over to the right side of the area on the 3 principles of heredity, and you will see a box where tall is indicated with a big T and short is indicated with a little t. Tall with a big T would represent the dominant version of the trait, and short with the little t would represent the recessive version of the trait. So if a parent has one of each, big T and little t, then that parent is going to be tall because the dominant version of the trait will hide or mask the recessive version of the trait. However, during the process of meiosis, when those gametes are formed, sperm or egg, some of the gametes are going to receive a big T from the parent, and some of the gametes are going to receive a little t from the parent. They separate during the process of meiosis. So at the bottom of that box, it asks, when does segregation happen? And you are going to answer during meiosis, when the gametes are formed. Mendel's last principle of heredity, which you will record in space for number 3, is the principle of independent assortment. Now you do need to write in this definition. You will have to write small and squeeze it in. Genes on chromosomes are separated independently of one another when the gametes are formed. This results in different combinations of genes in each gamete, allowing for genetic variation. So once again, this can be difficult to understand, so look very carefully at the diagram shown. In the first parent, there is a big A and a big B and a little b. Now, if you look way down at the bottom, where that parent has produced the gametes, 4 cells, you will see that some of them have a big A and a big B, and some of them have a little a and a little b. But the big A and the big B don't have to go together. Now let's look at the second parent cell, where they have the same letters, but they're arranged in a different order. There is a big A and a, then a little b and a big B. It's the same letters as in the first parent, but they've lined up in a different order. So in this situation in the gametes, looking at the bottom, some of them have a big A and a little b, and some of them have a little a and a big B. Even though both parents have the same letters, the same alleles to start with, they have different gametes produced because they have lined up, the chromosomes, have lined up in a different sequence. The big A and the big B don't have to go together. There are a few terms that are absolutely essential for you to know from memory when you are studying genetics and when you are doing genetics problems. There is a chart where you can write in these definitions. The term dominant, which we will represent with a capital letter, in this case we're going to use the letter T, is defined as a form of a trait that can hide or mask the other form. Recessive is represented by a lowercase letter, in this case a little t. It is defined as a form of a trait that can be hidden or masked. Allele is one of these two forms of a trait. We're using the letter T, so an allele would be either big T or little t, and it is defined as a form or a version of a trait. The word genotype is the combination of alleles that are present in an organism for a particular trait. So using the letter T, an individual might have a big T and a little t, or a big T and another big T, or a little t and another little t. The phenotype is the physical expression of the trait. So for example, if the genotype is big T, little t, or big T, the phenotype, the physical expression, would be tall. The term homozygous refers to having two of the same alleles for a trait, which is also called pure breeding because the parent can only pass on that allele to his or her offspring. So homozygous genotypes would be big T, or little t. On the other hand, heterozygous means having two different alleles for a trait, which is also called hybrid. This would be, as an example, big T, little t. Now look down at the bottom of your graphic organizer. You are asked, what is a trait? You are going to write in this definition here. A trait is a physical or a physiological characteristic of an organism. And traits are determined by your genes, or DNA, fill in the blank, which is a code for the production of protein, fill in the blank. So traits are things like your eye color, your hair color, whether or not you have dimples, whether or not you can roll your tongue. All of these types of traits, all of these types of characteristics are traits. Traits are also things like your blood type, a physiological characteristic, something that you can't see by looking at you, but which is something that is part of who you are. Now look over at the far right side of your graphic organizer. This is a section where we are going to learn about the idea of nature versus nurture. So first of all, fill in the blank for what is nature. In this context, when we use the word nature, that refers to your genes or your DNA. Nurture in this context refers to your environment. So this would be things like the types of food you eat, the education you receive, the medications that are available to you, your upbringing with your family. All of this is part of nurture. In humans and other organisms, diseases are often a result of nature, components in your DNA, and nurture, how you are taken care of. 3 examples of these diseases that you need to record in the space provided are cancers, diabetes, and heart disease. What this means is you may have a genetic predisposition for one of these traits. Just because you have a genetic predisposition for diabetes, as an example, does not mean you are necessarily going to get diabetes. However, if you do not eat right and exercise, you are more likely to develop diabetes. So it is part of your DNA, but it is also affected by how you are taken care of and how you take care of yourself. Nature and nurture and the effect of each on certain traits can be studied by using twins, and in particular identical twins, because they have identical DNA. So if we have twins who ideally had been separated at birth, we can study those twins and find out which characteristics they have in common and which characteristics they don't have in common because of their different upbringing. We're now going to be looking at the different types of inheritance or paths of inheritance that you've been studying in Module 8. Let's first of all summarize these along with the types of symbols that are used for each. There is a space to do this on the back side of your graphic organizer for Module 8. The first type of inheritance is normal Mendelian inheritance, so called because it follows Mendel's laws. The alleles for Mendelian inheritance are represented by 2 of the same letter, 1 capital and 1 lowercase. For example, big A, little a. The second type of inheritance is codominance, where the 2 alleles are equally dominant. They are represented by 2 different letters, both of them capital. For example, a big A and a big B. Incomplete dominance is a form of inheritance where neither allele is completely dominant. This is represented by 2 of the same capital letter, but one of them has the prime symbol. For example, big A prime. Multiple alleles is a type of inheritance where there are more than 2 possible alleles. For example, human blood type. In human blood type, we have 3 possible alleles. We have IA, IB, and little I. Sex-linked inheritance is also termed X-linked inheritance because this type of inheritance takes place when the alleles are carried on the X chromosome. In this type of inheritance, we have two possible alleles, both of which are carried on the X chromosome. For example, X big A and X little a. In polygenic inheritance, there is more than one gene controlling a trait. Poly is many, genic is genes. So in this case, instead of just having one gene controlling a trait, you might have a lot more than that. For example, gene A, gene B, and gene C. For the first type of inheritance, Mendelian, which is also simple dominant recessive inheritance, let's look at a sample problem. We have tongue rolling being dominant to non-tongue rolling. So we're going to show a cross between a homozygous dominant tongue roller and a non-tongue roller. In this cross, we have a big T being crossed with a little t. All of the resulting offspring are big T, little t. This means that all of the offspring are heterozygous, and the heterozygote can roll their tongue. So in the box for heterozygote, you need to write tongue rolling. You also need to complete the Punnett square as indicated. Big T for one parent, little t for the second parent. All of the offspring are big T, little t. The reason that the heterozygote can roll his or her tongue is because they have the dominant allele, the big T, which covers up or hides or masks the little t. The next type of inheritance is codominance. Remember, in this type of inheritance, we're using two different capital letters. So black feathers and white feathers in chickens are codominant. Show a cross between a black feathered chicken and a white feathered chicken. So parent 1 will be big B. Parent 2 will be big W. All of the offspring will be big B, big W, which is heterozygous. So complete the Punnett square as indicated. Then under the section for heterozygote, you will write black and white. These individuals that are heterozygous express both of the alleles because they are codominant, black and white. Incomplete dominance is different. Look back over, you will see that for incomplete dominance, you are using the same capital letter, one of which is assigned the prime symbol. In 4 o'clock flowers, red petals and white petals are incompletely dominant. Show a cross between a red 4 o'clock and a white 4 o'clock. So parent 1 is going to be big R. Parent 2 is going to be big R prime. All of the offspring are going to be big R prime. So fill in the Punnett square as indicated. Under the section for heterozygote, you need to indicate that all of the heterozygous flowers will be pink. Big R prime indicates that they are going to blend together. They won't be red and white. That would be a codominant type of inheritance. They will be pink, blended. The next example is for multiple alleles. And here we have the chart showing the different types of genotypes that are possible for various human blood types. So you first need to complete the chart. For blood type A, an individual might be IA, or IA little I. Having blood type B, an individual might be IB, or IB little I. In order to have blood type AB, an individual must have the genotype IA and IB. And finally, in order to have blood type O, an individual must have the genotype little I. So now let's show a cross between a man with type O blood and a woman with type AB blood. So parent 1 is going to be little I. Parent 2 is going to be IA, IB. Go ahead and set up your Punnett square and complete the Punnett square. You are then asked, what are the phenotypes of their children? So look in the Punnett square, look at the different genotypes that are possible. We have IA little I corresponding to blood type A, and we have IB little I corresponding to blood type B. So one parent with blood type O, one parent with blood type AB have the possible children with blood types A or B. For sex linkage, there are particular traits that you need to remember. There's a place to write an example. Color-blindness is one that often comes up. Don't forget, sex-linked is X-linked because it's on the X chromosome only. Please make sure that you fill in that blank. When we have a female who is heterozygous for a trait, she is said to be a carrier because although she doesn't have that particular trait, she could pass it on to her offspring. Because males only have one X chromosome, they show the trait more often. Make sure you have all of those blanks filled in. Now let's complete a sample Punnett square. We're going to show a cross between a man who has normal vision and a woman who is a carrier for color blindness. The genotype for the first parent is X big B Y. That's the male with normal vision, the dominant trait. The woman who is a carrier has the genotype X big B, X little b. She is normal, she has the dominant allele, but she carries the recessive allele. Go ahead and set up and complete your Punnett square. After doing this, you will see the possible offspring for the children. You might have X big B, a total normal girl, X big B, Y, a total normal boy. You might have X big B, X little b, a carrier girl, or X little b, Y, a colorblind boy. Next, we have polygenic traits. There's a place to fill in an example. Skin color is a polygenic trait. Remember, polygenic traits are often those that show a very wide range of phenotypes. Polygenic traits are governed by more than one gene, so we cannot do Punnett squares for these types of traits. But you can show the distribution of the traits graphically. This is called a normal distribution curve. Make some notes on the graph so that you understand what you're looking at. Most of the individuals are going to be average. That is your tallest bar in the middle. Go ahead and label that bar average. But there will be some individuals at either extreme. Some who have a very pale skin color and some who have a very dark skin color.